Hello everyone, it's Mr. Caudell. I hope you're doing well. I'm doing well. It's chapter 23.2. Here we go. Well, your book does some things terribly, and this is one of them. They take this pretty simple and super duper important topic and just butcher it. So I've told you in the I guess, notes on this chapter what to read and not read, and I don't really like anything that has to do with wildflowers with respect to this. As a result, we are going to be following this handout. Make sure that you get this. It should be on whatever learning management system I'm using. I should have a copy of this here for you. You should be handed out one of these in class if we're going to class together. It has some butterflies up here in the top right corner. Make sure that you have it, please. Well, the idea is this: we can use math to determine whether or not a population is evolving. Someone could just anecdotally say, oh, that population has changed over time or evolved or through natural selection. And then, of course, your answer always to them would be, prove it. So let's use math. It's called the Hardy-Weinberg equation. And it can be used to test whether a population is evolving. So what I've written here is the definition of a population. Because what is a population? Well, it's a localized group of individuals capable of interbreeding and producing fertile offspring. So what does that mean? Let me give you an example. Your book gives an example of this. These are caribou that live up in northern Alaska in the Brooks Range. And the point is that there's different populations in the same area. Now, these guys kind of live together, they interbreed, the range might overlap with another population, but that doesn't necessarily mean that they're going to strike out and make babies with the other individuals in the other population. It's just like people from the United States of America and people from Mongolia or people from Papua New Guinea. I don't know how to say it right. They're just different populations of people. They can interbreed. They're part of the same species. But it's not likely that they would, just generally speaking. But sure, it could happen. No problem. So that's a population. So let's go to this piece of paper. We're going to start in the upper left corner. It says, Darwin stressed that the members of a population vary, but he did not know how variations come about and how they are transmitted. It was not until the 1930s that population geneticists were able to apply the principles of genetics to populations and therefore develop a way to recognize when evolution has occurred. A population is all the members of a single species occupying a particular area at the same time. In population genetics, the various alleles at all the gene loci in all individuals make up the gene pool of the population. It's customary to describe the gene pool of a population in terms of gene frequencies. Suppose that in a Drosophila, that's a fruit fly population, 36% of the flies are homozygous dominant for long wings, 48% are heterozygous, and 16% are homozygous recessive for short wings. Therefore, in a population of 100 individuals, we have 36 big L, 48 big L, little l, and 16 little l, little l. Those are the genes or the alleles that exist in the population. What is the number of the allele big L and the allele little l in the population? So you can look at that math there. To determine the frequency of each allele, calculate its percentage from the total number of alleles in the population. In each case, for the dominant allele big L, 120 over 200 gives you 60%, or 0.6. For the recessive allele little l, you have 0.4. That means 60% of the alleles in the population are big L and 40% are little l. Sperm and eggs produced by this population will also contain these alleles in these frequencies. Assuming random mating, all possible gametes have an equal chance to combine with any other, we can calculate the ratio of genotypes in the next generation by using a Punnett square. There's an important difference between a Punnett square used for a cross between individuals and the following one. Below the sperm and eggs are those produced by members of the population, not those produced by a single male and female. As you can see, the frequency of the allele in the next generation is the product of the frequencies of the parental generation. Results of the Punnett square indicate that the frequency for each allele in the next generation is still 60% and 40%. Go ahead and take a look at that square there. Therefore, sexual reproduction alone cannot bring about a change in allele frequencies. Also, the dominant allele need not increase from one generation to the next. Dominance does not cause an allele to become a common allele. The potential constancy or equilibrium state of gene pool frequencies was independently recognized in 1908 by G.H. Hardy, an English mathematician, and W. Weinberg, a German physician. They used the binomial expression p squared plus 2pq plus q squared to calculate the genotypic and allele frequencies of a population. Figure 18.1 shows you how this is done. We're going to get to that in just a second. But first, let me give you an example. So, remember that the definition of evolution is a change in allele frequency in a population over time. Population is a group of interbreeding individuals. An example might be the Arrowhead elk population. Now, there are other elk populations in our valley. There's a Vail elk population, there's Eagle, there's one in Minturn. I'm sure that there's ones up behind the school up in Singletree up there. These are populations. They interbreed, they're together. Could they go and have a fun time with another population? Sure, and get pregnant and have babies. And of course, they're all part of the same species, but a population is such that they're kind of together most of the time. So let's imagine in the Arrowhead elk population, there's 100 individuals. That would mean that for 100 elk, there are 200 alleles for fur thickness. Let's imagine that on homologous chromosome number 5, in every elk, they got a chromosome 5 from their mom, chromosome 5 from their dad. When those two chromosomes come together in any cell, they have genes or alleles for fur thickness. Let's imagine this could be, don't you love it when they use letters that aren't that different from one another, capital and lowercase? So let's imagine that the big F allele creates a protein that creates thick fur, and little f causes thin fur to be made. So you could have homozygous dominant, homozygous recessive, or heterozygous like we're seeing here. Let's imagine that for 200 alleles for fur thickness, we see the big F happen 140 times and the little f happen 60 times. What then are the percentages of big F and little f in the population? You can do this math. This means that 70% of the genes in that Arrowhead elk population for fur thickness are big F, and 30% are little f. So what's important here is what we've established is that there's an allele frequency. That's what we've established here for these genes for fur thickness. Let's then imagine that we have certain environmental conditions and that those are pressurized, meaning these genes have naturally selective pressure on them, like if it's too hot or it's too cold. So let me show you this picture. So let's imagine that we have a cold snap and individuals that have lots of little f's, specifically a little f, which has given them a recessive condition where they have thin fur. Maybe they die and they don't pass along those genes. Would this gene frequency change? And it might. And I'm going to tell you, gene frequencies change all the time. And so populations are evolving all the time. Any organism that you look at and see, the owls and rodents and plants and everything, they're always under pressure, and certain things are advantageous and gene frequencies are changing. So the general idea is that at all the time, we see gene frequencies change and therefore evolution is occurring. So let's go back here and let's go to this figure 18.1 down here at the bottom. It's this kind of square down here at the bottom. We're going to do this calculation using the Hardy-Weinberg formula. And let me do this. I'm going to move the screen. Let's do it over here. So what I would like you to do is I'd like you to get out a fresh piece of paper. This absolutely is not a spectator sport here, doing this math in the evolution unit, because lots of kids say that this is the hardest part of the evolution unit. Just going to back this up just a Okay, so what I'd like you to write down at the top of your page is this: p squared plus 2pq plus q squared equals 1, and that p plus q equals 1. And I'll tell you what those algebraic letters mean in just a second. Just write that so you have that. You can also see that here in our square, down here at the bottom. This is well with my notes on it, but you can see it on your square as well. Basically, what this is showing us is that I just want to make sure that you're getting this, is that if you have big A individuals, and you have big A individuals, and you have little a individuals, if you take the frequencies of these genotypes, they have to add up to 100%. So the population is only going to have individuals with homologous chromosomes number 5 in every one of their cells in these types of combinations. Big F, little f, or little f. It's homozygous dominant, heterozygous, or homozygous recessive. That has to equal 100. And then if you take those individual frequencies like we did on the other board, where it was 0.7 or 70% big Fs for the alleles in the arrowhead out population for fur thickness, and 30% little f. So I'm just going to put here big A and little a. If you took the percentages of these alleles and how often they were found in the population's cells, it also has to add up to 100%. And so in order to do this problem, what I do every time, and I hope that you do this too, please do not cut corners on this, because I've seen smart kids miss these problems and they shouldn't have on tests. So just draw this. I draw this little matrix every single time I do one of these problems. And so I'm going to continue to fill this out. What I'm saying here is that it's just the same thing as those equations over there, is that the frequency of big A in the population plus the frequency of little a in the population has to equal 1, or 100% of the alleles that exist in the population. Because this is saying it's greater than 1. I'll just put an equal sign. Likewise, down here, if P is our algebraic representative for our large letter, which is the dominant allele in the cells of the population, then what is P squared? Well, that's just A. And what is Q squared? That's just little a. This one's a little tricky. P times Q is big A, little a. And so they had to, I get it, they had to put this coefficient in front, but we're going to let them deal with that. We're going to let them do that. And we're just going to put big A, little a. And what this represents is if you take the frequency, or how often, these are all decimals here that we're going to be dealing with. The frequency of big A, plus the frequency of the heterozygous condition for fur thickness or any other gene combination that we're looking at, and the frequency of the homozygous recessive or little a, you're going to get 100% again. So these also have to equal 1. So now, sorry, this is harder to do online than I thought it would be. Now that we've done that, we can do the math. So I'm down here at this box and it says, example. An investigator has determined by inspection that 16% of a human population has a recessive trait. I'm going to stop right there. I wouldn't go any further personally, because I know that if individuals in a population have a recessive phenotype or trait, what's their genotype? It must be recessive, or little letter, right? So before I go any further, I'm going to put what number right here? 0.16 or 16%. That's what this says, 16%. Now, okay, I'm not even going to read this, because let me show you how to do this. So if the frequency of the little a genotype in the population is 16%, what can we figure out here using this number? Well, if I took the square root of little a, what does that equal? Little a. But whatever I do to this side of the equal sign, let's put an equal sign here, I have to do to this side of the equal sign. Square root of q squared is q, and then I would have to square root this too. So what does that mean here? That means that 40% of the alleles in this population are little a. Well, since this has to equal 1, what does that mean about the big A frequency in the population? 60% of the time, right? Now let's figure out what's here. We know that all this has to equal 1. I'll get rid of this. What do you think we're going to do here with these numbers that are up above? Well, if we square A, that gives us big A, big A. If we square P, that gives us P squared. And if we square this, that gives us 36%. So 36% of the individuals in that fictitious population have homozygous dominant for the genes that create whatever trait we're looking at. And so because this has to equal 1, what must this equal? I can get that one of two ways. I can subtract all this from 1 and figure it out, or I can do 2 times P times Q. So what's 2 times 0.6 times 0.4? So this is 0.24 times 2 gives me 0.48. So what this is telling us is that 48% of the individuals in this fruit fly population on the piece of paper have heterozygous genotypes for whatever condition we're looking at. So let's figure this out. Now I'm going to go back to the question. It says, using this information, we can complete all the genotype and allele frequencies for the population provided the conditions for Hardy-Weinberg equilibrium are met. We haven't really gotten to that last little phrase there, but we can do all the math here. We did it. Okay, I usually have kids read this. Sorry that you're listening to me. Let's keep reading. I'm now up above the box here. It says, the Hardy-Weinberg principle states that in equilibrium of allele frequencies in a gene pool calculated by using the expression p squared plus 2pq plus q squared will remain in effect in each succeeding generation of a sexually reproducing population as long as 5 conditions are met. Go ahead and read those 5 conditions there. The Hardy-Weinberg principle. So here's the way that we're trying to get at this. It's almost like a null hypothesis in math. The idea is that you could say that a population is evolving, and if the definition of an evolving population is that their allele frequencies change over time, then let's set up a situation where there are no allele frequency changes over time. And if that then is violated, by default, we can say that the population has evolved. So what they've done here is they've set up this principle. They say, well, look at these 5 conditions: no mutations, no gene flow, no random mating, no genetic drift, and no selection by natural selection. If all those things happen, then there's going to be no change in the allele frequency over time. Fine, so no evolution. But the question is there ever an instance where this doesn't occur, where some of these are violated? And the answer is, of course. There's always going to be selective pressures, weather and food resources and the ability to run faster than another guy or predators. Is there always going to be random mating? No. Individuals are going to be stoked on a bird's plumage or a big rack on the head of an elk or whatever it may be. So that's not non-random. That's not random mating. That's non-random mating. Gene flow. There's always going to be genes coming from other populations. Like if the Vail elk herd guys want to mix it up with the Arrowhead ladies one night, and they do so, and then they cruise back on the bike path up to Vail. I don't know. Could that happen? Maybe. There's always going to be mutations. The point is that these things will always happen. You need to memorize these 5 things and understand exactly what they are. I'm almost done. In real life, these conditions are rarely, if ever, met, and allele frequencies in the gene pool of a population do change from one generation to the next. Therefore, evolution has occurred. The significance of Hardy-Weinberg principle is that it tells us what factors cause evolution, those that violate the conditions listed. Evolution can be detected by noting any deviation from a Hardy-Weinberg equilibrium of allele frequencies in the gene pool of a population. Does anyone have a question on this? This is fine, theory is good, but we have to be able to apply this to some scenarios here. And we will do a lab where we do that, where we change the environmental conditions in the class and we see if our population of individuals in the class evolves. It's really fun. I enjoy doing it every year. So do students. Okay. Sorry, I'm going to finish this. The accumulation of small changes in the gene pool over a relatively short period of 2 or more generations is called microevolution. Microevolution is involved in the origin of species to be discussed later in the chapter, as well as in the history of life recorded in the fossil record. Change in allele frequencies may result in a change in phenotype frequencies. Figure 18.2, the butterfly picture up here, illustrates a process called industrial melanism. Forced soot is introduced into the air due to industry. The original peppered moth population has only 10% dark-colored moths. When dark-colored moths rest on light trunks, they are seen and eaten by predatory birds. With the advent of industry, the trunks of trees darken, and the light-colored moths stand out and are eaten. The birds are acting as the selective agent, and microevolution occurs. The last generation observed has 80% dark-colored moths. So you can see that the phenotype has changed as a result of selective pressure. And by definition, the thing that guides the phenotype is the genotype. So the alleles in the genes must have changed over time as well. So has that population of these peppered moths evolved? Yes, if the allele frequencies change over time. A Hardy-Weinberg equilibrium provides a baseline by which to judge whether evolution has occurred. Any change in allele frequencies in the gene pool of a population signifies that evolution has occurred. This is much simpler than the way they do it in the text. We're going to do these 2 practice problems, 18.1, and then I'll shut down this lecture. So go ahead and read that and try it right now. I recommend on a fresh little area of the piece of paper that you reset this up. You got to set this up every single time. Please press pause and try this on your own. You probably don't want to look at his face, huh? So, although this one girl once went to college, it was a couple years ago, and she wrote me an email, and she said, I fall asleep with Mr. Anderson every night. Oh, what I mean is I watch his videos, and they put me right to sleep. I didn't know if that was a compliment or not. Anyway, whatever. So I guess you could watch, if you're having trouble falling asleep, just watch Mr. Anderson's videos. But I'm just kidding because I think that they're very good. That's what someone wrote me. Number 1 says, in a certain population, 21% are homozygous dominant, 49% are heterozygous, and 30% are homozygous recessive. So 30% are homozygous recessive. That means I can put a 0.30 here. Now, if I start square rooting this and then get a huge decimal here, a decimal there, if things look ugly, you probably have the wrong numbers in the wrong place. Something's wrong. I'm going to tell you, it used to be that kids were able to do this by hand, like in their head, without a calculator. So these are always going to be really nice. I'm not going to say whole numbers, decimal whole numbers, but it's got to look good. If you start getting like a decimal with 7 places or something, something's wrong. So stop and reevaluate at that time. I would get that here. So all of a sudden, something's wrong. So I'm going to keep reading. It says, what percentage of the next generation is predicted to be homozygous dominant, assuming a Hardy-Weinberg equilibrium? But of course, that's super easy. Why? Well, if we assume a Hardy-Weinberg equilibrium, that means that all of these conditions, these 5 principles, were met. That means that the population hasn't evolved or the allele frequencies haven't changed. So the answer, of course, in the next generation for homozygous dominant is 21%. No change. Tricky, huh? Go ahead and try number 2, and then I'll be done. Press pause if you need to. Of the members of a population of pea plants, 9% are short, recessive. I'm going to stop right there. 9% are short. If 9% of the population has a homozygous recessive phenotype, then I know that they have the homozygous recessive sorry, if they have the recessive phenotype, then I know they have the homozygous recessive genotype, little a, little a. I'm going to do the math here. If I square root this, I know that 30% of the alleles in that population are little a. That means 70% of the alleles in that population are big A. I could square everything here. 49% of the individuals in this population are homozygous dominant, big A, or whatever letters they're given to you, but I'm just using A's. And then I can do 2 times P times Q, that's 0.21 times 2 gives me 0.42, or 40%. 42% of the individuals in this population have heterozygous genotype. So let me finish. It says, what are the frequencies of the recessive allele little t and dominant allele big T? Well, we've got the answer here. What are the genotypic frequencies of this population? Here are the genotypic frequencies. Thanks everyone for your attention. We'll absolutely be doing more of these questions in class. If you need to, ask me for help. Look in the book. Mr. Anderson has a really good video on this stuff that might be able to help you. Thanks for your attention.